Now, we don't have that many problems in Chapter 17, right? Only 2 problems, plus, plus a couple more from the microRNA study, but not too many. And I think these are pretty easy. So 17.14, present an overview of RNA-induced gene silencing achieved through RNA interference, so Rnai. How do the silencing processes begin, and what major components participate? Okay, so what do we need to know for this? What I would like you guys to focus on for Rnai, and you should also look at how microRNAs are generated, but that's not part of this problem right here. So for Rnai, we are somehow ending up with double-stranded RNA in the cell. And I should point out here, we talked a little bit about co-suppression in plants with petunia and quelling of canidia pigment color, canidia color or spore color in Neurospora, before we went on to sense and antisense silencing. All of these processes now are thought to essentially work through double-stranded RNA-induced RNA interference. So this is a double-stranded RNA. So this co-suppression and quelling, even though it involved DNA, through some complicated or confusing explanations, it seems by the way these transgenes integrate, they can actually give rise to transcripts that make double-stranded RNA. So that's a little aside if you were wondering how co-suppression and quelling fit into our whole talk of RNA interference. The sense and antisense was pretty straightforward. So you can go back and look at the slides about those if you are interested. But for this question here, we are just focused on the mechanism of RNAi, the general mechanism. So it starts with a long double-stranded RNA. And when I say long, I mean longer than the small interfering RNAs that are going to be produced in a moment here. So somehow double-stranded RNA enters the cytoplasm, and that's processed by an enzyme called dicer. And dicer chops up the RNA into little pieces. Let's just say 21 base pairs long. And these are called siRNAs, for small interfering RNAs. So dicer chops these up into little pieces, and then these are used by the RISC complex, RNA-induced silencing complex, and I'm going to put a couple of proteins in here. So there are a lot of proteins involved in this complex, and they don't actually look like a circle, a triangle, and a square, but I'm just, indicating different proteins. So one strand of the small RNA is going to be incorporated into the RISC complex. Now this is going to be like the targeting mechanism. Targeting mechanism. I have no clue if there should be 2 T's there, so I'm not even going to put a second T there. I'm going to guess that's how you spell targeting. Okay, so it's the targeting mechanism, and it's going to search for messenger RNAs that have a complementary sequence here. So here's a messenger RNA. It could be much longer than this. And if this small RNA is complementary to the part 21 nucleotide segment of the messenger RNA, if that is true, it'll be cut. It'll be cut in half. And I'm not actually sure the siRNA is cut. You don't need to know that either. But the messenger RNA will be cut. It's a 5' cap. The 3' end's going to have a poly A tail, right? So it'll be cleaved, and then these guys will be degraded by exonucleases. into individual ribonucleotides. So that's the overall mechanism of RNA-induced RNA silencing or RNA interference. So what is the point here? Well, one point is, well, this messenger RNA that's just been destroyed cannot be translated into a protein. So not translated into a protein. So it's a mechanism of gene silencing. So that gene was being transcribed, but before it could be translated, or before it could be translated again, if it's already been translated into a protein, it was destroyed by RNA interference. So messenger RNAs can be destroyed, and sometimes, especially with microRNAs, it's thought that they aren't destroyed, but somehow translation is just blocked. So a ribosome can bind to the messenger RNA, or maybe it can't even bind because the RISC complex has identified it and dragged the messenger RNA to a place where ribosomes can't get to it. And so in some of our figures from our PowerPoint, you're going to see that we have this 3' UTR. And some of our microRNAs, which are also incorporated into the RISC complex, for some reason, a lot of them seem to be specific for 3' UTRs of their target genes. So we have the RISC complex here, binding to a 3' UTR of the target gene. And there's a lot of evidence to say that in many cases that these messenger RNAs aren't destroyed, but we know translation rates decrease. So somehow this identification of the 3' UTR with the incorporated microRNA doesn't always lead to destruction of the messenger RNA, but it somehow prevents translation. So again, we can have translational suppression during RNA interference, especially if it involves microRNAs, or RNA cleavage, especially if it involves siRNAs. Okay, so if you know either RNA cleavage, messenger RNA cleavage, or translational suppression, both of those are good enough for understanding or demonstrating how RNA interference works.